Good morning, good afternoon, or good evening. This is Lisa Whitehead, and this is Biology 205, Anatomy and Physiology 1 at Illinois Central College. Today, we're going to be looking at Chapter 25, reviewing metabolism, nutrition, and energetics. Hold on to your hats. There are a lot of learning outcomes for this chapter. I highly recommend you take advantage of having these to use these as your study guide. So you'll be looking at defining metabolism, anabolism, and catabolism, and its role in utilizing molecules for the cell. In the process, we'll review endergonic and exergonic associations for each. We'll describe what oxidation and reduction reactions, also known as redox, are and how they're used in metabolism. We'll discuss how to write the equation for aerobic cellular respiration. We'll describe the role of the coenzymes NAD and FADH in cellular respiration. We'll distinguish between aerobic and anaerobic respiration processes and the benefits of each. We'll discuss why glycolysis occurs in the cytoplasm and why that's an advantage. And then we'll also discuss what it is exactly that occurs during the process of glycolysis. So we'll describe redox reactions, the process and quantity of ATP, how it's utilized and produced, which is total net gain, by substrate level phosphorylation. We'll describe the process and quantity of NADH plus hydrogen ions and FADH2 and how that's produced. We'll describe the process and quantity of carbon dioxide being produced, of water being produced, and we're going to be able to list the carbon molecule that is entering and leaving glycolysis, and describe lactic acid fermentation and the Cori cycle. Also, we'll explain the preparatory reaction and transition step and pyruvate oxidation, which includes discussing where the step occurs and explain why it requires oxygen. We'll describe the redox reactions and describe the process and quantity of ATP that's utilized and produced, which is total net gain, by substrate level phosphorylation. We'll describe the process and quantity of NADH plus hydrogen ions and FADH2 that's produced, the quantity of carbon dioxide that's produced, of water that's produced. We'll list the carbon molecule that is entering and leaving this step, and explain why this reaction happens twice per molecule of glucose. We will also explain what occurs during the TCA or Krebs cycle or citric acid cycle, also known as the Cori cycle. And in this process, we'll discuss where the step occurs and explain why it requires oxygen. We'll describe the redox reactions. We'll describe the process and quantity of ATP that's utilized and produced. Again, this is total net gain by substrate level phosphorylation. We'll describe the process and quantity of all of these things being produced. I won't list again. We'll be listing the carbon molecule that is entering and leaving this step, explain why the reaction happens twice per molecule of glucose, and know how ATP is produced by substrate level phosphorylation. Next, we're going to look at the electron transport chain. So we'll discuss where this occurs, why it requires oxygen. We'll describe redox reactions and the process and quantity of ATP that's used and produced, and that's all by substrate level phosphorylation. We'll also talk about the ATP that's used and produced by oxidative phosphorylation. We'll describe the process and quantity of all of these molecules again, and then we'll list any carbon molecule that is entering or leaving this step, and describe what cytochromes are and what their function is in this step. And then lastly, we will list the energy yields per NADH plus hydrogen and FADH2. We will summarize the energy yield of glycolysis and cellular respiration. We'll describe how efficient this process is and where the lost energy goes to. We'll discuss the processes of gluconeogenesis, glycogenesis, glycogenolysis, and lipolysis. We'll also identify triglycerides as the most abundant lipid in the body. We'll describe how during lipid catabolism, how one molecule of glycerol, which is pyruvic acid, and 3 fatty acid molecules are going to enter the TCA or Krebs or citric acid cycle and why they require oxygen to do so. We'll also look at comparing the energy benefits of using lipids as opposed to carbohydrates as an energy source. So then we'll discuss several reasons why we don't use lipids over carbohydrates as a source of energy. And we'll explain why there are non-essential and essential fatty acids in lipid synthesis. Last slide of learning outcomes, I promise. Lastly, we will look at discussing how almost any organic substance can become a lipid via the process of lipogenesis. We'll also explain how each amino acid enters the TCA cycle at different places and then how the energy benefit is going to differ accordingly. We'll explain the general process of transamination and deamination of amino acids. We'll explain several reasons why we don't use proteins over carbohydrates as an energy source. We'll compare what are non-essential versus essential amino acids and know what a normal body temperature is, both in Fahrenheit and in Celsius. Also, we'll describe the homeostatic mechanisms that maintain a constant body temperature. We'll explain the process of heat exchange within the environment. So in this process, we'll compare radiation, conduction, convection, and evaporation. And last, we will explain which of these processes is the most effective given normal circumstances.
As you can see in this lecture outline, we're going to review some material that isn't part of Chapter 25 this week. Because we're omitting sections 5 and 6 from Chapter 25, it gives us a little bit of time to insert a little bit of a refresher. So we're going to go back and look at the section on enzymes from week 2, which is covered in Chapter 2, specifically sections Chapter 2-3 and 4. And we're just going to go through this kind of briefly as a refresher. The enzyme component will be covered in your lab, but it won't be covered in this second exam in the lecture component of the course. That said, this is an area that's sometimes difficult for students, so it's a nice opportunity for us to review, so that way you'll be a little bit stronger in this area when you have to take care of this on the final cumulative exam. So first we're going to discuss enzymes, and then we'll move on into Chapter 25.
The function of an enzyme is just to lower the activation energy of a chemical reaction. So for example, what that means is that you don't need as much heat to trigger a chemical reaction. But that said, what they're really doing is functioning as a catalyst. And catalysts are amazing because what they do is they speed up a chemical reaction without themselves being changed or consumed. So that is really important. So substrates have to bind to a specific region on the membrane, and that region is called the active site. And so the tertiary or quaternary structure of the enzyme is what's going to determine what the shape is of the active site. And keep in mind that there are different enzymes that are required to metabolize each isomer. So we have a whole number of different enzymes that are involved. In future labs, you're going to be working with enzymes to be able to assess how they affect the rate of a reaction. Now we already know that the function of an enzyme is to lower the activation energy of a chemical reaction, but which factors affect it in which direction? So here is a list. So if you decrease the temperature, you're going to decrease the rate of a reaction, right? This makes sense because we're slowing down molecular movement with decreased temperature. But then as we increase temperature, we're going to actually increase the rate of the reaction occurring, which makes sense because with heat, we're going to have more molecules moving around more quickly. So that would facilitate molecules running into one another more frequently, which would then result in the rate going up. Also, when you have an increased temperature, if it's too high, you can denature the proteins. Further, if you lower or raise the pH, that can decrease the rate because for the most part, most reactions have an ideal pH. So any deviation from it is going to totally ruin everything and denature your proteins. If you increase the reactant concentration, that will probably increase the rate of your reaction. And also, if you increase the enzyme concentration, then you have more workers to help get the work done, right? The enzymes are doing the heavy lifting of speeding things up. So with more of those workers, you will get more done sooner. So that increases the rate.
So just to reiterate the information from the previous slide, there are a very specific set of circumstances that each enzyme has in which they operate best. For example, you might be a morning person and I might be a night person. So enzymes have their own specific preferences, and they're really not going to do their job outside of those preferences. So temperature is very important. If the temperature is too high, they denature. The proteins denature, and therefore it won't work at all. And if the temperature is too low, it just slows down the reaction time dramatically. Also, pH is another factor in which there's an optimum range, and that range for each reaction is very narrow. So any deviation from that is going to totally change your outcome. And that optimum range is going to vary by enzyme. Now, if you consider the next 2 points, concentration of enzymes and concentration of substrates, follow me with this example of imagining construction workers trying to build a house. So the enzyme is basically workers trying to get the job done. So if you have more workers trying to build a house, that's going to get the job done a whole lot more quickly than if you only have 2 people doing it, for example. And the same thing is true. So if you're building a really big house, you have more substrates. So there's more work to do, so it's going to take longer. So these are all important factors in determining the rate of enzyme activity.
This video really helps to clarify how enzymes work. So take a moment to watch this and follow the link, and then come back to where you were in the audio.
Figure 223 in your text shows you generally how substrates will bind to active sites of an enzyme. So starting on the left in section 1, you'll see the enzyme down below having two active sites. So the two substrates, S1 and S2, fit perfectly into those active sites. So in step 2, you'll see that those two substrates are now bound to the active site, and so they're interacting with one another. Once this is all together as one giant piece, it's now called the enzyme-substrate complex. And so now the substrate is going to be bound together, and that binding is going to alter the shape of the enzyme. And so that is what allows the new product to join together. And then in step 4, our product, which is the combination of S1 and S2 from image 1, is able to be joined together and to be spent out from the enzyme, and the enzyme is now ready to restart the process again with two new substrates. Here's a quick review of some vocabulary terms from our previous lecture. So enzymes are biological catalysts that remain unchanged in a reaction. So we just saw in the previous image that the enzyme was the blue blob at the bottom, which is responsible for pulling together the two substrates and then spitting them out together as one individual product. So there's a lower activation energy required for a chemical reaction with the presence of enzymes. Again, activation energy is just the energy required for the reaction to happen. Now, coenzymes and cofactors are very similar in that they're both required for a reaction to occur, but coenzymes are typically water-soluble vitamins, whereas cofactors are actually metal ions. Now, substrates are going to be the reactant or whatever it is that goes into the reaction. So from the previous slide, those were the two yellow blobs at the top that S1 and S2 were the labels on them. And then the product is when you've bound that S1 and S2 together. So now they are a joined substance. So that is the material that comes out of a reaction that an enzyme has facilitated. And an indicator is just any test at all that will verify for you the presence or the absence of a reaction. So an example of an indicator would be something like a litmus test. In your lab, it's going to be helpful for you to understand that there are different enzymatic models by which enzymes do their work. So the first is the lock and key model, which is kind of what we looked at earlier, and the induced fit model is another alternative. And so in both cases, the enzymes are doing the same thing. They're a catalyst facilitating a reaction to two substrates that are floating along outside of the enzyme. So how they work is one of two ways. So the lock and key model basically means that the substrates, which are the orange triangle and then the light blue rounded shape on the top example, those have to fit exactly. And if they don't fit exactly, they're rejected by the enzyme, and the enzyme will not actually perform its action. Now, it's a little different on the induced fit model. If you look down below in the lower example, you'll see the bright green rounded substrate and the bright green triangular substrate right beside it. Those don't match exactly if you look at the corresponding substrate right below it. The active site is a little bit off. But when those two substrates come to the enzyme and approach the active site, it actually forces a change of the shape of the enzyme so that it accepts those two substrates and can then either form a bond between the two of them or break bonds, but either way it's able to create its catalytic reaction and produce the product that leaves the enzyme to go off and do their own jobs. So you can see that they're totally different processes, but keep in mind, just because the image on the top shows you a product that is two substances together, the two substrates are joined, and in the bottom image it shows you two separate products, that is not to say that in the lock and key hypothesis we are only creating bonds, and in the induced fit model we are only breaking bonds. We can do either with both of these two hypotheses. This is just an example schematic, so that way it tries to make this make a little more sense. In today's lecture, we're going to be talking about a lot of different chemicals, and so I just want to review with you some of the terminology for naming compounds. So if you've taken medical terminology before, you've probably already got this, but just in case you haven't, or just in case you've forgotten, I just want to go through this very quickly. So when you see terms that end with ACE, ASE, that is an enzyme. So for example, lactase is going to be an enzyme that digests lactose. For example, if you are lactose intolerant, you would take lactase. I think the actual brand name is lactase or something like that, but that's basically the exact same thing. We're showing that it is an enzyme. If something ends in ate, that's going to be an acid that is an ionized form. So Ringer's lactate is going to be something that you'll use as an IV solution, and it's a byproduct of lactate metabolism. So that'll be used to counter any fluid loss if it's related to an electrolyte imbalance. You'll study this much more when you get into your nursing courses. And then anything that ends in ose is a sugar. So for example, ribose, lactose, fructose, sucrose, these are all sugars. And again, somebody could be lactose intolerant, so they have to take the lactase enzyme to help them to digest the lactose that's present in milk products. There are all sorts of enzymes. Today we're only going to study a small number of them that are present in our body. But just to give you some ideas of what we're about to be talking about, kinase is going to be an enzyme that's going to add a phosphorus group. And so if you have an enzyme that's going to add phosphorus groups together, there's going to be its exact counterpart, right? One that removes phosphorus groups. So in this case, we have phosphatase, which removes the phosphorus groups. We also have hydrolase, which is going to split water, and so it'll add hydrogen ions and hydroxide ion groups during decomposition reactions. There's also dehydrogenase, which will remove hydrogen again. So it dehydrogenates, so it takes away hydrogen. And isomerase is going to rearrange atoms within a molecule because it's basically creating an isomer, right? So the example that you see on the left is an image of glucose and fructose. So they have the same atoms, but they're just moved around in a different configuration. So shape matters for enzymes and in chemistry in general. Here are some more specific enzymes that you're likely to run into. So the first is carbonic anhydrase, and this is extremely important in blood. So what happens is this is going to be responsible for converting carbon dioxide into carbonic acid, which can be transported by red blood cells, and then takes it back to carbon dioxide for exhalation. So this is an incredibly important enzyme in the role of us getting adequate gas exchange. Amylase is also another specific enzyme that we need to look at, and it's important in saliva. So you're going to be doing some testing with this in your labs, and it's a lot of fun. But basically what it's going to do is be produced in your pancreas and your salivary glands, and it helps us to digest carbohydrates. And what are carbohydrates? Sugars, right? So we'll talk more about that in the lab. And then the last one listed here is catalase, which is an important part of lysosomes. What it does is it protects the cell from oxidative damage by reactive oxygen species. So remember, those lysosomes are a site of hydrogen peroxide and also peroxisomes as well. And so that can be very damaging to the surrounding structures within the cell and all of the organelles. So this catalase in the lysosomes is a very important component of protecting the cell. So in summary, some of the characteristics of enzymes that are really important to recall are specificity, competition, and saturation. And what I mean by that is, in specificity, each enzyme has a specific fit that it allows for the substrate that approaches it. So without that match, there is going to be no reaction between the substrate and the enzyme. Also, competition is another component. So if there are molecules with a similar shape, it's possible that somebody else, when I say somebody else, I mean another substrate, might come in to the active site first. So if there's competition, they may rule out the full number of reactions occurring for the specific substrate in question. And then lastly, saturation. And what this just means is if all of the active sites are occupied, then that's it. There's no more work that can be done. We cannot speed up the reaction if all of the workers are actively working. We can't make them go any quicker. The pace goes at the pace that it goes. Pop quiz time. The area on the enzyme to which a substrate will attach is the A, product, B, active site, C, lock and key, or D, competition site. If you recall from previous slides, we looked at how an enzyme will have two different substrates approach the active site. So the active site is the point at which they will attach. What comes out of the reaction is the product, and that can either be bonds being built or broken. So those are the two different models I showed you in both the lock and key model and the induced fit model, in which it demonstrates both breaking down and bonding together two different substrates. And then competition site seems like it would be a very good option to answer this question with, because there is in fact competition among the various substrates to attach to the active site. but the active site is truly the name on the enzyme of where the substrate will attach. So the answer is B. The change of shape that occurs in an enzyme after substrate attachment is called A, competition, B, saturation, C, specificity, or D, induced fit. Now, options A, B, and C are all important elements of how an enzyme will be able to function, whether all the different active sites are filled so that way it's totally saturated, or if it's only going to fit a certain shape, so that's specificity, or for competition, which is option A, if you have two different enzymes of the same shape trying to fit into the active site. But what actually happens when the enzyme changes shape around the substrate that approaches the active site is the induced fit model, which is different than the active site on the lock and key model, in which case the shape is exactly the same and the enzyme shape does not change to accommodate the incoming substrate. So the answer is D, induced fit. Alright, so that was our review on enzymes. Now we're going to start with chapter 25, and again recall that we are studying only chapter 25, sections 1 through 4 and 7. So let's first go into metabolism overview, in which we look at just the general functions of catabolism, anabolism, energetics, oxidation, and reduction. Section 25-1 in your text gives you an overview of metabolism in general, and it gives you some terms to learn that are going to be helpful for you as we move into the actual processes that drive how we manage to process all sorts of organic molecules, ranging from lipids to sugars, etc., etc. So the term metabolism is very broad. Essentially what it means is that this includes all of the chemical and all of the physical changes in your body tissues as we both break down and build up new organic molecules. So when we break down organic molecules, we use the term catabolism. And in catabolism, we have a number of different subtypes depending on what exactly it is that we're breaking down. So if we're breaking down lipids, we use a process called lipolysis. If we're breaking down glycogen, the process is called glycogenolysis, right? We're breaking down glycogen into glucose. And then, of course, we have to break down glucose, right? So that's called glycolysis, with the lysis, of course, being the loosening or the breaking down. And then the first part of the word, our root, is telling us what exactly is being broken down. So in glycolysis, glucose is being broken down into pyruvate, which we'll discuss in more detail in subsequent slides. Anabolism is exactly the counter opposite of catabolism. So instead of breaking down organic molecules, we are building them up. And a quick way to remember the difference between catabolism and anabolism is think about anabolic steroids, right? Those are for building up big muscles. So anabolic is building. And catabolism, I think of a catastrophe. Everything is falling apart. It's breaking down. So that's the way I keep it straight in my mind. So anabolism is really important because we synthesize new organic molecules all of the time, and this is important in creating structural repair for our tissues, allowing us to grow. Also, so that way our secretory cells can create different types of secretions, and further, it allows us to store nutrient reserves in times of either famine or just times of lack of a certain nutrient in our diets. Now, a nutrient pool is just the body of all of the different types of nutrient molecules that are present in our blood. So what this means is if, for example, a certain cell requires copper and there is not an adequate amount of copper in our nutrient pool in our blood, that nutrient pool will not have it in order to be able to provide it to the cell, and therefore it is absent. So we can only draw from the nutrient pool what is actually in it. Figure 25-1 in your text shows you how we metabolize all of these organic nutrients and then how this creates nutrient pools for our body to draw from. So it's kind of a complicated image, but we'll walk through it step by step. So starting at the top, you'll see that we have our diet. Everything that we consume is included in this category. So that includes fats, carbohydrates, and proteins. So starting off on the far right with proteins. When you consume a protein, it will be broken down into amino acids. And then those amino acids will be part of the amino acid pool from which your body can draw. So we can either synthesize protein from that amino acid pool, or if there's an excess of amino acids, then it will go over into the glucose pool over to the left in the very center of the image. So this process is called gluconeogenesis. So let's go back up to the top and look at carbohydrates now and how that works its way down. So when you consume carbohydrates like bread, rice, etc., those all get broken down into sugars, and specifically glucose. So as it gets broken down to glucose, you'll see that there are two different options. So the product of glucose can either go down into the glucose pool, so that way we can use it in our bodies, or if there's an excess, then it's going to shift over to the left into the fat stores. So now this is an interesting point. So when the excess glucose goes over to the fat stores, what we're talking about here are adipocytes, so all of the fat tissue in your body. So let's go back up to the top again of fats. So when you consume fats, which are required for us to absorb a lot of different nutrients like A, D, E, and K, and also just for general function of many different parts of our tissues, those will be broken down into either free fatty acids or glycerol. And then from there, they'll either be used for metabolism in most tissues, or alternatively, they can be broken down into a free fatty acid pool, and then by a process called lipogenesis, will go up into the fat stores to be stored for later. So as you can see, there are a couple different mechanisms for excess nutrients. So down at the very bottom, you'll see there are two green holders. So on the right, we have brain metabolism, and that is feeding right off of the glucose pool. And the reason for that is that the brain has a very high glucose need. So for that reason, neurons, or the functional unit of the brain, are going to be very sensitive to any lack of glucose. And then on the left, you'll see that metabolism in most tissues is labeled. And that's because most of our tissues are going to take energy from both the glucose pool and also from the free fatty acid pool and our fat stores, which is another way to get energy. But I'd like you to note something right away. So you'll see the metabolism of most tissues and your brain metabolism, both at the bottom. These both draw from fats and carbohydrates. There's no fast direct route between the proteins that you eat into supporting the metabolism of most of the tissues in your body. In order for this to happen, when you consume proteins, proteins have to be broken down into amino acids. They have to go into the amino acid pool, and then in order to be able to be used, they're going to have to go through that process of gluconeogenesis, so that way it can be made into glucose so that it can be taken up by the body. We'll come more into this topic a little bit later on, but I just wanted to give you the broad 30,000 foot view before we get into the details. So I'm sure that you all remember that mitochondria are the powerhouse of the cell, right? They provide energy in the form of ATP, or adenosine triphosphate, for all of our cellular operations. Now, the way that they do that, though, is kind of interesting because the mitochondria are what you might call picky eaters. They won't just take up anything from the intracellular fluid called the cytosol. They're only going to take in very specific things. So the cell has to feed the mitochondria from the nutrient pool that is in the extracellular fluid, so that way it can feed the mitochondria what it will in fact eat. So things in the nutrient pool, again, will include things like water, various vitamins, mineral ions, and organic substrates, which include carbohydrates, lipids, etc., etc. So let's talk about mitochondria a little bit more. So the chemical reactions that occur in the cytosol are essentially just breaking down nutrients, that way the mitochondria can take them in. So when you see the letter M here, I'm just abbreviating mitochondria. So when the mitochondria breaks down nutrients, it's going to break them down into carbon dioxide, water, and ATP. That is the main goal of mitochondria. So carbon dioxide and water are two byproducts, but the main purpose of mitochondria is to generate adenosine triphosphate. And the mitochondria do this by a process called oxidative phosphorylation. And so oxidation is just the oxidation of molecules from the citric acid cycle, which we'll discuss coming up, and phosphorylation, which is turning ADP, or adenosine diphosphate, into adenosine triphosphate. So together, the oxidation and the phosphorylation is known as oxidative phosphorylation. Energetics is simply the study of the flow of energy and its transformation. So as we discussed previously, the brain, for example, has a very high glucose requirement. And different tissue types all have their own specific energy and nutrient requirements. So that varies from tissue to tissue. So for example, neurons require glucose, but skeletal muscle, when it's actively contracting and performing work, is going to require a lot of oxygen. And these requirements don't just vary according to tissue type, they also vary according to demand. So, for example, think of your skeletal muscle. When you're just sitting on the couch watching Netflix, Your skeletal muscle isn't working very hard. But when you go out for a jog, of course, you're using a lot of energy, and those muscles are going to demand a lot of oxygen. So the need for that specific substance is going to go up according to exercise. Similarly, when you're sleeping, you're resting, versus when you're awake, you're moving around more. So a good example of this would be your brain requiring glucose, or also, honestly, just your skeletal muscle again. And then further, as we grow, when we're young children, we have a lot more metabolic need for a vast majority of our tissues. So growth is very demanding as compared to maintenance, which is the stage most of us are in once we're over the age of 20-25. Now let's discuss oxidation and reduction. And I know that this can be very intimidating because I was very intimidated by this when I studied this the first time. So let's walk through it, and we'll take a 30,000-foot view, and I'll give you some tips on how to remember this, and it will make sense, I assure you. So what we're discussing here in oxidation and reduction are energy changes during a transfer of either oxygen, hydrogen, or electrons. And this occurs during any chemical reaction or bond. So if you'll recall, back in Chapter 2, we talked about covalent bonds and ionic bonds. And you'll remember covalent bonds were sharing electrons, whereas the ionic bonds were the giving or receiving of the very specific electrons. Well, that's exactly what's happening here, okay? So if we have two molecules having an interaction and they're going to either bond or break up, somebody is going to gain an electron and somebody is going to lose one. That's it. That's what we're talking about here. So yes, it also has an implication for oxygen and hydrogen, but primarily what we're talking about are the electrons and where they go during a reaction. So when you think that one of the molecules is going to give up their electron, that is going to be called oxidizing, or we're going to say that it went through the process of oxidation. And so because it lost an electron, it loses some potential chemical energy. Now, the opposite is true for the recipient of who gets that electron. That molecule is going to have been reduced or went through a process called reduction. And this is going to result in an increase of potential chemical energy because it just gained another electron. Okay, so that's it. Very simple, very straightforward. So you'll see at the top of the screen, I put electron transfer -oil rig. This is in your text too, and this is a very simple way for you to remember what is happening with oxidation and reduction, because reduction sounds like the opposite of what you're going to think. So oxidation is loss and reduction is gain is what the acronym oil rig stands for. So think of this all of the time as you're studying this, and it will help you very much. But keep in mind, when we're talking about oxidation is loss and reduction is gain, we are specifically referring to electrons, okay? But there is more than just what's happening in a reaction than electrons, but for purposes of this class, that's what we're going to be discussing, just the electron transfer to or from a different molecule, okay? So you can almost forget about the gain or loss of hydrogen or oxygen. Do study this so you understand it, but in terms of remembering which is oxidation and which is reduction, just focus on the electrons. And so now keep in mind that these reactions are always paired. So that's why they're called redox or reduction reactions. So what's happening here is we're having one molecule give up an electron, the other one receives it. So because of that, one element is going to be oxidized, the other is going to be reduced. Hence, this is together called a redox reaction. The electron transport chain is really just a series of protein complexes through which mitochondria are going to pass electrons. So as they pass the electrons from protein complex to protein complex to protein complex, they're going to be changing the protein complexes along the way. Okay? And it does this through the process of reduction reactions or redox reactions. And so the final electron acceptor at the end of the process is water. So we have a byproduct of water and then whatever other protein complex the mitochondria is creating. Coenzymes are an important factor in this process because they act as an intermediary for electron transfer. And what you can imagine in your head is 3 basketball players standing in a row with one basketball player passing it to the guy in the middle and then the guy in the middle quickly passing it off to somebody who's going to go and take a shot. And that's basically the idea. The coenzyme is going to hold the ball or the electron just temporarily and then pass it off. So they're an important part of the play, but they're not themselves actually creating the protein. They're part of the assembly line. So a couple different examples of coenzymes that are going to come up over and over through this chapter are NAD, or nicotinamide adenine dinucleotide, and flavin adenine dinucleotide, which is also called FAD. And both of these will remove hydrogen atoms. And so hydrogen in its state is going to be just really a hydrogen proton and an electron, right? So when a coenzyme accepts a hydrogen atom, that coenzyme is now reduced and gains energy. Let's go back to reduce, because reduce sounds like lesser, right? But remember, oil rig, oxidation is loss, reduction is gain. So when we're saying the coenzyme is reduced, it actually is gaining an electron, and hence it gains energy. Now the donor molecule that gave the electron to the coenzyme is oxidized, right? Because it lost that electron and it loses energy. So these two coenzymes, which are also known as carriers, the NAD plus and FAD, they are both going to be responsible for transferring over electrons, and they're going to do it slightly differently. So the NAD, or nicotinamide adenine dinucleotide, is going to be able to bind 2 electrons and 1 proton. Okay? Now contrast that with FAD, the flavin adenine dinucleotide, which is riboflavin or vitamin B2, is going to be able to bind 2 electrons and 2 protons. So FAD can do a little bit more heavy lifting than the NAD+, but they're both still very important coenzymes in this process. Okay, pop quiz time again. As NAD gains electrons as part of whole hydrogen atoms, NAD is blank to NADH. Okay, so that sounds complicated, but let's stop for a second. So the question is asking you, as NAD plus gains electrons, NAD plus is what to NADH? Okay, so if it's gaining electrons, go back to oil rig, right? Oxidation is loss, reduction is gain. So if NAD plus is gaining electrons, it is reduced. So the answer is C, reduced. Which of the following can denature an enzyme? A, low temperature, B, high temperature, C, lower than optimal pH, D, higher than optimal pH, or E, all except A can denature an enzyme. Well, this is a tricky question because the question is asking you which can denature an enzyme. We're not talking about what can make the enzyme process slower. We're talking about denaturing it fully, right? So we know that heat for sure will denature a protein, right? We boil an egg and that denatures the proteins in the egg and we get a hard-boiled egg as opposed to a raw egg. So we can rule out B. We know that that can denature an enzyme. But what about everything else? Because we have to figure out, are any of the other options appropriate, considering option E says all except A. So lower than optimal pH and higher than optimal pH can denature an enzyme, but low temperature won't denature it. It just won't function as quickly. So the answer is E. All except A can denature an enzyme. The breakdown of glycogen to produce glucose for metabolism is A, gluconeogenesis, B, glycogenesis, C, glycogenolysis, or D, glycolysis. Now, you don't even need to understand these processes in full to be able to answer this question correctly, because what you really want to do here is just break down the constituent word parts so that way you can rule out some options. So we know that genesis is the generation or the creation of something. So we can rule out right away option A and option B because both of those are genesis or building up reactions rather than breakdown reactions. So that leaves us with options C and D, which both end with lysis, which means loosening or breakdown. So we're on the right track. So what you need to do here is differentiate between glycogenolysis and glycolysis. And what's important here is to recall that the question is asking you how to break down glycogen, not what the end product is. So if we're breaking down glycogen, the process is glycogenolysis, which is the answer. But if we were breaking down glucose, then the answer would be D, glycolysis. But for this question, the answer is C, glycogenolysis. All right, so now we've reviewed enzymes and we've had the 30,000-foot view of the metabolism overview. Now we're going to move into the details of how we metabolize carbohydrates. So this is going to include glycolysis and aerobic metabolism functions, including the citric acid cycle and the electron transport chain. carbohydrate metabolism really just circles around glucose. Glucose is the easiest thing to break down to get ATP and to get it quickly. So you can see why this would be very important for a lot of our cells. So glucose catabolism is the breakdown of glucose, and it allows us to produce ATP, or adenosine triphosphate. And through this process, we go through a series of steps. The first is glycolysis, then the citric acid cycle, then the electron transport chain, and finally chemiosmosis. Now, you're going to probably imagine there are times in our bodies when we don't have adequate glucose. And this is really a problem because many of our tissues are highly dependent on glucose, for example, the neurons in our brain. So in that case, we need to generate glucose. So we build it up anabolically. So the glucose anabolism processes include gluconeogenesis and glycogenesis. So both of those terms end in genesis, so we know they are generating or creating new. So gluconeogenesis is creating new glucose, and glycogenesis is creating glycogen. And altogether, this process is called cellular respiration. This is really the sum of all these enzymatic processes that are going to allow us to extract energy from the things we eat, specifically the organic substrates like carbohydrates, fats, and proteins. Okay, so more specifically, what is glycolysis? Well, glycolysis is glucose catabolism, or the breakdown of glucose. And this process is always anaerobic, okay? Please note this because this is the only part of cellular respiration that we're going to discuss that is anaerobic. And here's how you'll remember that's always anaerobic. This is the only part that occurs in the cytosol, which is the fluid in the cytoplasm, right? So because it's happening there, it does not include oxygen and therefore is anaerobic. All the other steps are going to occur in the mitochondria. So when we're doing glycolysis, we are just breaking down glucose into pyruvic acid in the form of pyruvate ions, which can be useful in further steps of breakdown. So in order for glycolysis to occur, we're going to need to have all of the following. Glucose, of course, that's our starting point. We have to have enzymes in the cytosol that can break down the glucose. We need to have ADP and ATP, inorganic phosphate groups, and NAD. If any of those are missing, glycolysis cannot occur no matter what. So all of those are absolutely mandatory components to allow the glycolysis process to occur. There are actually a couple different examples here that are interesting. So one is red blood cells. So they get all of their ATP only from glycolysis, which is really kind of neat. So this is an important process for our red blood cells to get their energy. And then also, skeletal muscle is going to use glycolysis during times of strenuous activity as well. Here's a quick summary of glycolysis, which is really just glucose catabolism or glucose breakdown. And again, this is happening anaerobically in the cytosol, right, in preparation for the citric acid cycle. So we start off with a single glucose molecule, which we'll call a 6-carbon glucose molecule, and it enters this pathway of glycolysis. So it's formed by an enzyme which will phosphorylate a glucose molecule, creating glucose 6-phosphate. So that costs 1 ATP molecule in order to phosphorylate that single glucose molecule to make it glucose 6-phosphate. Now a second phosphorylation will occur, and this is going to cost a second ATP molecule. So the outcome of this is that we have 2 3-carbon pyruvate molecules formed by phosphorylation, and the final outcome is that we have 2 net ATP molecules produced. And when I say net, I mean overall. So for example, if you were to invest $2 of your own into the stock market, and when you took the money out, you got $4 back, you really only made $2, right? Because 2 up front were yours to begin with. So that's the idea here. 2 net ATP are produced, even though 2 other ATPs were already spent in the process of phosphorylating the glucose molecule. So after this, we have 2 pairs of electrons that are passed to 2 NAD, forming 2 NADH. And so the overall summary of glycolysis, and this is important, is that 1 glucose molecule gets broken down into 2 pyruvates and 2 ATPs. That is the outcome of glycolysis. So understanding that, in summary, glycolysis is 1 glucose molecule converting to 2 pyruvates and 2 ATPs, we understand that the 2 ATPs are energy units, but what happens with the 2 pyruvates? Well, either the oxygen supply is going to be inadequate, and therefore the pyruvate will be reduced to lactate, and the NAD will be oxidized from the NADH. Now, just remember, reduced and oxidized, oil rig, oxidation is loss, reduction is gain, right? Or, if the oxygen supply is adequate, then the mitochondria is able to absorb the pyruvate and will be able to generate ATP for cellular activities. And why is this? Because oxygen is required from every step beyond glycolysis because it happens in the mitochondria. So in this case, the mitochondrial outer membrane will be permeable to ions of small organic molecules, and the pyruvate fits that category. So these pyruvates are able to enter the mitochondrial intermembrane space, and then mitochondrial carrier proteins are going to move the pyruvate into the mitochondrial matrix. Now, all of this is in preparation for the citric acid cycle and then the electron transport chain. We have now arrived at the citric acid cycle, which is a cycle known by many names. It is known as the citric acid cycle, the Krebs cycle, because it's named after the person Krebs who identified this, the TCA cycle, also the tricarboxylic cycle. But citric acid is the very first substrate of the citric acid cycle, so this is becoming the name that's used most frequently. So we'll refer to it as the citric acid cycle for the sake of simplification for the remainder of this course and also through 206. So this cycle occurs in the mitochondrial matrix aerobically, requiring oxygen, and starts with pyruvate. Green means go. That's why I have this in green up top. Okay, so we start with pyruvate, which is our outcome product of glycolysis. The purpose of the citric acid cycle is just to remove hydrogen ions from organic molecules and then to transfer them to coenzymes. That's it. That's the purpose. So pyruvate is going to react with NAD and coenzyme A to yield one carbon dioxide molecule, one NADH, and one acetyl-CoA, which is just a 2-carbon acetyl group. So this is the systematic breakdown of carbon, oxygen, and hydrogen bonds. You're going to see the term decarboxylation a lot, and what this means if you break it down is D, we're taking something out, carb is carbon, oxy is oxygen. So we are removing carbon and oxygen and then releasing it as carbon dioxide in decarboxylation. Now the product, the overall outcome of the citric acid cycle is as follows. So we start with one pyruvate molecule and it gives us 3 carbon dioxides, 3 waters, 4 NADHs, 1 NADH2, and 1 GTP, which is guanine triphosphate, which is really just the equivalent of adenosine triphosphate. Still remember that it's GTP, not ATP, but it functions as ATP. Oxidative phosphorylation is just the process of generating ATP from transferring electrons from coenzymes to oxygen through a sequence of electron carriers. And this all occurs in the mitochondria. Oxidative phosphorylation is really important because it produces over 90% of the ATP used by our body cells. So if oxidative phosphorylation was ever to be stopped in any way, the cells will die. That's it. So oxidative phosphorylation is that important. Now cells are going to be able to get their oxygen just by diffusion from the extracellular fluid, right? And without oxygen in the extracellular fluid, or without adequate oxygen in the extracellular fluid, mitochondrial ATP production stops. And when that stops, we have no oxidative phosphorylation and we have cell death. The outcome of oxidative phosphorylation is water, right? So we have 2 hydrogens and oxygen which come together to give us 2 waters, and that's it. So this is a very powerful reaction, and so we have to slow it down. So that's why we have all of these electron transfers happening over and over and over through these redox reactions of electrons being both oxidized and reduced. And this all occurs in the electron transport chain coming up next. The electron transport chain is embedded in the inner mitochondrial membrane. Okay, so keep in mind as we're breaking down glucose, we go from the cytosol, where we have glycolysis anaerobically. We go to the outer aspect of the mitochondria, where now we're in the mitochondria. It is now an aerobic process, and we have the citric acid cycle. And once we are in the inner mitochondrial membrane, we have the electron transport chain. So we're moving into the center of the mitochondria. Again, this is an aerobic process. So this just really consists of 4 respiratory protein complexes: coenzyme Q and cytochromes, which we'll talk about cytochromes more in pathophysiology for sure. And a cytochrome is just a protein plus a pigment, and that pigment is going to include either iron or copper. Proton pumps are going to be responsible for moving hydrogen along, and the formation of ATP is going to occur as electrons and protons are going to be moved to lower energy levels through oxidative phosphorylation. So again, we're using oxidative phosphorylation because we want to have a slower step-by-step process rather than one big burst. So for each NADH, we're going to get 3 ATP molecules, and for each FADH2, we're going to get 2 ATP. Figure 25.4 in your text goes into detail about the electron transport chain, but on the left are my quick notes of how to summarize what's happening in this process. So again, the electron transport chain is occurring in the inner mitochondrial membrane through an aerobic process, and let's start. So NADH is going to pass electrons from the respiratory complex to coenzyme Q. Now, energy from the electrons is going to be able to pump the protons into the intermembrane space. So the electrons are going to go to the respiratory complex 2 and then to coenzyme Q. And then the electrons will go to respiratory complex 3, and energy from these electrons will now pump protons into the intermembrane space. The electrons go now to respiratory complex 4. I know this is getting a little bit long-winded. The energy from this is going to pump the protons into the intermembrane space, and the electrons are going to be released to the matrix to form water. So finally, hydrogen ions are going to be able to enter the matrix through channels, providing energy for ATP synthase, which is an enzyme to build ATP, and it does that by combining ADP and P. So adenosine diphosphate plus another phosphate equals adenosine triphosphate, or adenosine with 3 phosphates. Chemiosmosis is also called chemiosmotic phosphorylation. And really all this is creating a concentration gradient across membranes. So this is a form of potential energy. So we have both the chemical component and an osmotic component. So it's going to support ATP formation indirectly. It doesn't cause it. This is not an enzyme. But what it does is it creates a steep concentration gradient across that mitochondrial membrane specifically, so that energy from electrons passing from site to site is going to be able to power those hydrogen pumps. And those pumps are what moves the hydrogen from the matrix into the intermembrane space. And those pumps also create a large electrochemical gradient. So all of this together provides energy essentially just for the process of converting ADP to ATP. So what is the overall outcome of metabolizing one molecule of glucose through glycolysis, which is anaerobic, and aerobic metabolism, which includes the citric acid cycle and the electron transport chain? So here's what we have. We get 2 ATP from glycolysis directly because we have glucose broken down anaerobically, and that creates 2 pyruvate molecules. We also get 3 to 5 ATP from the NADH that's generated in glycolysis. We get 2 ATP molecules from the citric acid cycle. So both revolutions each yield 1 ATP, or in the form of GTP. And then finally, it's the electron transport chain, which is going to create the vast majority of ATP through this process. The electron transport chain will give us 23 molecules of ATP, and it does this by shuffling from the citric acid cycle to the electron transport chain. So in summary, through metabolizing one glucose molecule through glycolysis and then through also the citric acid cycle and electron transport chain, we will end up with a total of 30 to 32 molecules of ATP. Please study this page and be sure that you're comfortable with knowing this information. So we've used some of these terms previously, but I still want to go through them with you here because we'll be discussing glucose anabolism, which is the building up or creating of glucose. So glycogen is really just the way that we store glucose in our liver and skeletal muscle. So I think of when I go on massive backpacking trips with my husband, how I'm burning up my glycogen stores in my liver because I am burning so much energy in my body. And then glycogenesis is when we get glycogen from excess glucose. Okay? And so that can then be stored in the liver and skeletal muscle. Glycogenolysis is the breakdown of glycogen to glucose monomers, so again with the lysis being the breakdown. And then gluconeogenesis is the formation of new glucose. Genesis is generation, right? And this is a totally separate process, by the way, because once glycolysis is completed, we can't just reverse it. So gluconeogenesis is an entirely new chemical process. Pop quiz time. How many net ATP are generated during glycolysis? A, 0, B, 1, C, 2, D, 4, or E, 32. Now this question is asking you how many ATP are generated during glycolysis specifically. So this does not include the electron transport chain and the citric acid cycle. So you can rule out 32 because 32 would be our overall largest possible value. So now we're down to options A through D. So we're looking at net, right? Now we know we have to spend ATP in order to get ATP, but in the end, the final result is that we will only have 2 ATP generated net. So the answer is C, 2. How many net ATP are generated during the citric acid cycle? A, 0, B, 1, C, 2, D, 4, or E, 32. Well, we're discussing here only the citric acid cycle, not glycolysis and not the electron transport chain. So because we're just discussing the citric acid cycle, the answer is C, 2. Because each revolution of the citric acid cycle will yield 1 molecule of ATP. So in total, we have 2. How many net ATP are generated by substrate level phosphorylation during the electron transport process? A, 0, B, 2, C, 23, or D, 32? Well, we're discussing here only during the electron transport process, which is the component of the whole entire process of breaking down one glucose molecule that will create the most. So 32 would be the answer that would indicate all of the ATP generated, and 2 is how much is how much is created by the citric acid cycle and also by glycolysis. And 0 is obviously not correct. So the answer is C. 23 net ATP are generated during the electron transport process. Where are ATP molecules generated during anaerobic metabolism? A, glycolysis, B, acetyl-CoA formation, C, the citric acid cycle, or D, electron transport chain? So this question is asking you, where are the ATP molecules generated during anaerobic metabolism? And so as we know from reviewing before, that the processes of the citric acid cycle and electron transport, et cetera, are all aerobic. The only part of the breakdown of glucose that results in an anaerobic process is glycolysis. So the answer is A, glycolysis. How many carbon dioxide molecules are generated during aerobic metabolism? A, 0, B, 2, C, 4, D, 30, or E, 32? Well, we know that overall, in the whole entire breakdown of one glucose molecule, we'll have about 32 CO2 molecules in total. But that is not to say that the entire process is aerobic, because glycolysis at the very beginning is an anaerobic process occurring in the cytosol. So therefore, we have to subtract whatever is generated in that process from the overall number of ATP developed through the breakdown of one glucose molecule. So if 32 is the total number of ATPs that can be generated through the whole entire process, and we know that 2 ATPs are created net through the anaerobic process of glycolysis, we take 32 minus 2 to end up with the answer D, 30. Where are water molecules generated during aerobic metabolism? A, glycolysis, B, acetyl-CoA formation, C, the citric acid cycle, or D, electron transport? Well, first off, the tip is aerobic metabolism, so we can rule out glycolysis right away. So now you're down to 3. And if we look at where water is generated, we end up with one water molecule that gets provided through the electron transport chain. So the answer is D, the electron transport chain. Now let's look at the way lipid is metabolized. So we're going to review section 25-3 in your text, which will review lipid catabolism, the beta oxidation of fatty acids, lipogenesis, storage, and distribution. Next, let's look at lipid catabolism, or how lipids are broken down. So the primary process that we'll look at is lipolysis, which is the breakdown of fats, and they'll be broken down into glycerol and or fatty acids and or monoglycerides. And then once they're broken down, one of two things can happen. They can either be converted into pyruvic acid, or they can be channeled directly into the citric acid cycle. Hydrolysis is going to be involved here in that it splits triglyceride into component parts. So of course we're breaking down water, right? And so we'll end up with one molecule of glycerol and 3 fatty acid molecules. Enzymes that are present in the cytosol are going to convert glycerol into pyruvic acid, and so then the pyruvic acid is able to enter the citric acid cycle because pyruvic acid is the end of glycolysis and is the beginning of the citric acid cycle in general. So pyruvic acid is the end of lipid catabolism through the process of lipolysis, and pyruvic acid is also the end of glycolysis. There are different enzymes that will convert fatty acids to acetyl-CoA, and this process is called beta oxidation. Beta oxidation is a process that is going to break down fatty acid molecules into 2 carbon acetic acid fragments, and FAD and NAD are reduced. Again, remember oil rig. Oxidation is loss of electrons, reduction is gain. So they've gained electrons. This occurs inside the mitochondria and is important because it's basically a part of the process that creates acetyl-CoA directly. So at each step, we're really just generating acetyl-CoA and NADH and leaving a 2C carbon chain bound to coenzyme A. Lipogenesis is essentially the development of fat, the building of fat. This is an anabolic process. So we are synthesizing lipids. This occurs in diets high in carbohydrates and also several hormones, for example, insulin. Now, lipids can be synthesized from almost any organic substance. So if you remember that very first image where it shows you the nutrient pool of the carbohydrates, lipids, and proteins, and how basically everything can become a lipid, right? Because if you eat too much of anything, whether it's protein, carbohydrates, or lipids, it all can be stored for future energy as fat in our adipocytes. So this includes lipids, amino acids, and carbs. They can all be converted to acetyl-CoA. Now, essential fatty acids are those that cannot be synthesized by the human body, so we have to ingest them. But these are required to synthesize prostaglandins in our body, so these have far-ranging consequences. So 2 examples of essential fatty acids that we cannot synthesize that we must ingest are linoleic acid and linolenic acid. And both of these are present in a lot of seeds and nuts and legumes, so you can easily supplement your diet just by eating these sorts of foods healthily and in moderation. Lipids are an important energy reserve for us because the breakdown of lipids yields a lot of ATP. So these are stored as water-insoluble droplets in the cytoplasm of cells. And these water-insoluble droplets, if you think about it, make sense because if you think of butter, butter is basically a liquid drop of fat, and that's what you can imagine these droplets being similar to. So you can imagine if these droplets in the cytoplasm are large, then the enzymes, which are water-soluble, are going to have a difficult time lysing them, which is part of why it takes a little more time and energy to break down lipids as opposed to breaking down glucose. So lipids can provide a lot of ATP, but they do it slowly, so this is not a quick energy source. So if you are an athlete, you need access to quick glucose, not necessarily lipids, because the lipids are going to take a lot longer to break down to provide energy. Lipids are also processed in the mitochondria, so therefore they require oxygen, and this is aerobic metabolism. And the cholesterol that we have will circulate in our bloodstream, and it circulates as a lipoprotein. We're going to discuss this more in the next slide in just a second. Also, free fatty acids, which are typically bound to albumin, are going to diffuse very easily across plasma membranes. So this is important in that when they cross the plasma membrane, then they can allow other different cellular activities to occur following that. Most lipids circulate through the bloodstream as lipoproteins, and this includes cholesterol. So lipoproteins are going to contain large, insoluble glycerides and cholesterol. Again, when we think of fats, think of drops of butter. So of course they're going to be water insoluble, right? So this includes things like chylomicrons. Chylomicrons are the largest of the lipoproteins, and they're produced by the intestinal epithelium deriving from things that we consume in our diet, from dietary fats. So what they do is they will absorb lipids from the food that is inside our intestinal tract, and then they will transfer it into the lymphatic system, which then gets passed into our bloodstream. The liver is the primary source of the remaining groups of proteins, and I'm sure you've heard of all of these before. So the first is very low density lipoproteins, or VLDLs. And these are responsible for transporting triglycerides to skeletal muscle. Low density lipoproteins are what may be known as the bad cholesterol. And they're bad because what they do is deliver the cholesterol to the peripheral tissues. So this type of fat is basically forming arterial plaques in the form of atherosclerosis, and that can have a number of sequelae, ranging from occluding blood vessels and therefore causing somebody to, for example, in the case of a diabetic, potentially have an occluded vessel behind their knee, which results in gangrene and the need for an amputation. Or if that arterial plaque lands in the blood vessels of the heart, it can result in the occlusion of one of the coronary arteries, and then that can create a myocardial infarction or heart attack. Now, high-density lipoproteins are the good cholesterols that you've probably heard of before, and what these are responsible for is for transporting any excess cholesterol from the peripheral tissues back to the liver. An interesting clinical correlation listed in your text is the clinical note discussing blood testing for fat. And really what we're discussing here is cholesterol blood work. And you've probably had several people in your family have this testing done already. So what this is just a simple blood draw in which we look at the circulating triglycerides in the blood to be able to determine what types they are. So we want the total cholesterol value, but we also want to break down what the amounts are of high-density lipoproteins, low-density lipoproteins, and very low-density lipoproteins. So because these different lipoproteins have different functions, it's important to know the levels of each. So somebody comes in to get this testing done, and they're expected to have been fasting. And if they are, then a normal range would be between 40 to 150 milligrams per deciliter. If the person has not fasted, and let's say they just went and had a cheeseburger and french fries, they're going to have a significantly higher blood triglyceride level, which is artifactually higher. So it's hard to interpret what the values are. So it is very important that the person truly be compliant and fast prior to this blood work. So when we look at the breakdown of the blood work, we're going to see all these different values. And previously, when this testing was new, we originally looked at just the total level of cholesterol, period, not really looking at all of the individual subtypes of lipoproteins. So if somebody had high cholesterol, that was bad. If it was low, that was good. And that was it. But that's not the way we look at it anymore, because now we understand the importance of the different roles of these different lipoproteins. So, for example, if somebody has a high cholesterol rate overall, and their low density lipoprotein is high, that's considered to be the highest risk, because the low density lipoproteins are the quote unquote bad cholesterols, which deliver cholesterol out to the peripheral tissues and therefore can become arterial plaques and clog up blood vessels. And then if somebody has a high cholesterol level, but their total cholesterol is normal, but their high density lipoprotein is low, that can also be suggestive of high risk, because it suggests that we're not basically handling that cholesterol that's being brought back to the liver adequately. So all of these factors will come into play to determine if somebody's truly at high risk or not. But ideally, we would like to see in healthy individuals lower overall high cholesterol. And if they do have anything higher, we would like to see a higher high density lipoprotein and a lower low density lipoprotein. Let's discuss section 25-4, protein metabolism. We're going to look at amino acid catabolism, transamination, deamination, and protein synthesis. Amino acid catabolism can be considered the last-ditch effort of the body to create energy when glucose and fats are low. So there are 20 amino acids that are going to comprise 100 to 140,000 proteins that are all synthesized by our body. And they do this by active transport and growth hormone affecting the way they can transfer across the plasma membranes. So these amino acids will be synthesized into enzymes, hormones, structural elements, and neurotransmitters, so they have a very important role. But prior to engaging in the citric acid cycle, these proteins have to go through several different things, which is part of why they're a slower process. The first is transamination. So this is really just removing an amino group and transferring it to a keto acid. They also have to undergo deamination. So we're deaminating, so we're removing an amino group. And liver cells are the primary site for where this takes place. And then the urea cycle has to happen as well. And this is producing urea from ammonium ions that have occurred from the deamination process. And the urea gets excreted in urine. So urea is composed of urine. Now, these are really difficult bonds to break. And the ammonia that's produced from the deamination is very toxic to cells. So this is a difficult process to undergo. The ammonium being toxic is really important to keep in mind, because if that's the case, then the presence of it is going to threaten homeostasis. So if we ever undergo extensive amino acid catabolism, we're risking jeopardizing the homeostasis of our body. So proteins in general are structural and functional for our body. So think about proteins as being part of our muscles. As soon as you start breaking down things like proteins and muscle, that's obviously not ideal. What we want to break down first is sugar, second back up is fat, and then third finally is protein. And if you want to try to think of a way to remember this, I think of it in terms of starvation, right? You eat a meal, you'll first process the sugars that you just absorbed from that meal. And if you don't eat for, let's say, a day, you start breaking down body fat. And if you don't eat for a week, when you're out of those reserves, your body will start processing muscle and protein as a last-ditch resort to have some sort of energy to keep things going. So hold that in your mind as we go through a little more discussion about proteins. So regarding protein synthesis, there are 10 essential amino acids that we have to obtain from our diet because we cannot synthesize them. Now, 2 of them actually can be synthesized, which is arginine and histidine, but we don't create enough of them, particularly for young growing children. So we count all 10 of these essential amino acids as being essential because we cannot synthesize them ourselves. Now, non-essential amino acids are those which our bodies can synthesize on demand. And they do this through amination, in which an amino group is added to use ammonium ions as a reactant. Pop quiz time again. How many ATP can be produced in the citric acid cycle? A0, B1, C2, D4, or E32? I'm hoping right now that 100% of you listening have selected the correct answer, which is C2. Where would a 3-carbon glycerol molecule enter the metabolic process? A, during glycolysis, B, during acetyl-CoA formation, C, during the citric acid cycle, or D, during electron transport? If you flip back through your notes, you'll note that during glycolysis, we break a 6-carbon glycerol molecule into 2 3-carbon glycerol molecules. So the answer is A, during glycolysis. How many ATP may be produced during the complete aerobic metabolism of one glucose molecule? A2, B4, C16, D30, or E38? So keep in mind this question is looking at the complete aerobic metabolism. So if it is the aerobic metabolism, that's ruling out any anaerobic metabolism. And we know that glycolysis is an anaerobic process. So if you were to look at the complete metabolism of one glucose molecule, including glycolysis, we would have a figure of between 30 and 32. So if you subtract 2 from that, you're going to get a figure of 28 to 30. So if you look at what your options are here, you can easily rule out all of the options with the exception of D, which is 30. After covering sections 25-1 through 4, we're going to skip sections 5 and 6 and move into 25-7, which is metabolic rate and thermoregulation. In this section, we're going to discuss units of measurement, the basal metabolic rate, heat exchange, and mechanisms of heat loss and generation. When we discuss metabolic rate, we're going to talk about calories and how they're burned. And this is an important distinction because you're going to read in a lot of different publications, ranging from magazines to books, that they'll discuss calories. And calorie may or may not be capitalized, but they're referring to the capital C calorie here, and I'll explain in a second. So in general, the way that we come to understand how we measure energy is energy that is required to raise the temperature of 1 gram of water by 1 degree Celsius is a calorie. Okay, but this is the lowercase c. The energy required to raise the temperature of 1 kilogram of water 1 degree Celsius is a kilocalorie, or the capital C calorie. And this is what we're typically referring to. So if you were to imagine that an average human weighing roughly 150 pounds, that would turn out to be about 68 kilograms, right? So when we do the math to figure out how many calories of the capital C that person should consume daily to maintain their weight, we would use the capital C for kilocalorie. We wouldn't think of the average human as weighing 68,000 grams and then give them some enormous number of grams of food they can eat in a calorie measurement with the lower C. So just keep in mind when we talk about calories for us, we typically mean capital C kilocalories. To measure the energy content of the different types of foods we eat, we use something called calorimetry, which is just measuring the total energy that's released when the bonds of the organic molecules are broken. Again, the organic molecules are the carbohydrates, the lipids, and proteins. And so this is determined by burning food with oxygen and water in a calorimeter, which excludes any other external interference with its measurement. So what has been known is that lipids will release just over 9 kilocalories per gram of energy. Carbohydrates will release 4.18 kilocalories per gram, and proteins are similar, just over 4.32 kilocalories per gram. So it's interesting to note that lipids release almost double the amount of energy that both carbohydrates and proteins do. Let's discuss the difference between metabolic rate and your basal metabolic rate. So the metabolic rate is just your average caloric expenditure. So it is the average amount of kilocalories that you're going to burn daily, just depending on what you do. So for example, one day you might go for a walk, the next day you don't, but on average you're going to have a metabolic rate that hovers, let's say, around 2,000 calories a day. Now, the basal metabolic rate is going to be just the basic amount of energy that your body expends at rest just to keep all basic functions alive. So this might be somewhere in the neighborhood of, let's say, 1,500 kilocalories per day. There will be energy gains and losses, of course. Some days you eat a little more. Sometimes you burn more calories through more exercise. So energy is going to be released whenever those chemical bonds are broken, and that energy will be used to synthesize ATP in cells. Keep in mind that some energy will be lost as heat, so we're going to discuss thermal regulation coming up shortly. And all of this is affected broadly by the amount of exercise somebody gets, their age, because their metabolism can slow down as we get older, sex also, because hormones circulating in our body are part of this and affect how we metabolize our calories, and also the climate in which you live. So if you're living somewhere very cold, your body has to keep warm, so it actually creates more heat and burns more energy as opposed to if you're somewhere warm. So all of these factors can come into play in terms of determining how your metabolism rates. When we discuss the regulation of energy intake, what we're really talking about is appetite control. Why you might be more hungry one day than another, and there are so many factors that come into play with this. Some of them are chemical, some of them are situational. So for example, if you're out with friends and everybody's having snacks at a Super Bowl party, you may enjoy more snacks than normal, for example. It may be in some way affected by mood. If you're feeling sad, you might eat more, or if you're feeling very sad, you might not eat very much at all. So emotion can come into play. If you've been more or less active, if you feel like it's hot out and you just don't feel like eating and maybe a salad seems better than a heavy meal. So there are tons of factors, but there are chemical factors that have been identified in recent years that are worth discussing here first. So thyroxine is one that is going to control our overall metabolism, and this is derived from our thyroid gland, which is in our neck. There's also a substance called cholecystokinin, which is abbreviated CCK. And so cholecystokinin and adrenocorticotropic hormone, which is from our adrenal glands, specifically from the cortex of the adrenal glands, hence adrenocortico, those are both going to be responsible for suppressing appetite and making you feel like you're full, you just don't want to eat anymore. Leptin is going to be released by adipose tissues during the absorptive state. So that will bind to CNS neurons, and that suppresses appetite too. So you'll see some articles in different magazines that will say like leptin is the thin hormone. Leptin thin, I guess that makes sense. Ghrelin, I've always thought of your stomach growling. So ghrelin is released by an empty stomach, and that stimulates your appetite and makes you want to eat more. So all of these are factors. But like I said, this is a multifactorial concept, but it is worth knowing that there are a whole variety of different reasons why our energy intake may be regulated irregularly. Obesity is only discussed briefly in this chapter. In fact, there's only one quick paragraph about it, but you will discuss it in much more detail when you take pathophysiology, and indeed in your clinical training. This is going to come up again and again. Obesity is an epidemic in this country, and there are a whole host of associated diseases, including heart disease, cancer, and diabetes, and these all result in people having a lower quality of life and a shorter lifespan. We define obesity as a body weight that is more than 20% above the ideal weight for a given individual. Thermoregulation, although it seems like this might fit into a chapter about skin, is actually best fit into this chapter about metabolism because heat is a byproduct of metabolism. And anytime you have increased metabolic activity, for example, if you go for a run, you're going to generate more heat. And that heat in your body is going to be retained by water. And water, keep in mind, holds onto heat quite well, which is why a swimming pool will actually stay warmer than the surrounding air on a cold night for quite a while. So for body temperature to remain constant, we have to constantly be losing some heat to the environment because we're constantly generating heat as a byproduct of our metabolism. So our body controls are going to measure heat gains and losses and adjust accordingly in order to maintain homeostasis. And we need to make sure that we maintain this homeostasis because the enzymes in our body only have a very narrow temperature range in which they will operate. So if things get too hot or too cold, they stop operating properly. And in fact, if they get too hot, we denature the proteins. Figure 25-13 in your text shows you all four of the main types of heat exchange with the environment. There's radiation, convection, evaporation, and conduction. Now, if you are a cook or a baker, you're probably familiar with some of these, but we are going to discuss each of these in a little more detail in the subsequent slides. Radiation is how objects that are warmer than the environment lose heat. So when you think about the sun shining down on you, that is radiating heat to you. So heat is being lost from the sun and you are feeling it. And this is what accounts for more than 50% of our heat loss while we're indoors. Just heat rising up out of our bodies. This is different than convection, and these two can be confused easily. So convection requires air movement to flow across the body surface, and when that air movement goes by, it helps heat to rise away from the body, and then cooler air comes back in its place. So keep in mind that heat generally rises in a house, so the upstairs might be warmer in winter, provided that it's well insulated. This accounts for about 15% of all of our indoor heat loss. Also, by the way, if you're a baker, you'll know that convection ovens have a fan in them, and so it moves the heat around more quickly, and so that actually helps to brown and cook things a little differently than through typical radiation ovens. We also have conduction, and conduction is the direct transfer of energy through physical contact. So the image that we just looked at of the woman doing a yoga pose, she had her hands and feet on the ground. So if you put your hands and feet on cold ground, you will lose heat through your hands and feet directly into the ground. This is not an effective mechanism of heat gain or loss in order to maintain our homeostasis, but it does have an effect, and it certainly has a perceptive effect. Hence, if you put socks on in winter, you might feel a little bit better than if you're walking around barefoot on a cold floor. Evaporation is the last mechanism of heat exchange. This absorbs energy. So when we perspire, you'll find that there is moisture on your skin, and once it evaporates, then it cools the surface underneath. So this is an important cooling mechanism. And the rates of evaporation on skin are really variable. Recall from previous lectures talking about skin, there is insensible and sensible perspiration. So insensible perspiration is when water crosses the epithelia and evaporates, and it can account for up to 20% of our indoor heat loss. And we don't even notice this. This is just as we're sitting here right now, this is happening. Sensible perspiration occurs from the sweat glands and occurs specifically usually when you're exercising or feel stimulated in some way, like if you're nervous before an exam. And so for that reason, sensible perspiration amounts are going to vary widely. The hypothalamus, which is an organ in your brain, is responsible for regulating our heat gain and our heat loss. So it coordinates all of our heat gain and loss through the heat gain center and the heat loss center in the preoptic area of the anterior hypothalamus. When we cover the central nervous system later on, this will all come back to you. And so this is an important site because it modifies the activities of other hypothalamic nuclei. Nuclei are just basically like centers. We're not talking about a nucleus specifically in regards to a cell. So temperature control can be achieved by regulating a couple different things, either the rate that we produce heat at or the rate at which we lose heat to the environment. But we can always modify these. So we can have behavioral modifications, for example, dressing differently. My husband is always warm, so he wears shorts when it's like 65 degrees out, and I'm wearing a jacket and jeans and shoes and a hat. So we make behavioral modifications to adjust what our natural sense of heat and cold perception is. Let's discuss how we can both increase our heat loss and conserve heat. Because remember, you can't gain cold. You can only gain or lose heat. So if we want to increase our heat loss to cool down, what happens is when our preoptic area in our hypothalamus senses that a certain set point has been exceeded, then our heat loss center gets stimulated. And this does a number of things. It inhibits the vasomotor center, and that causes peripheral vasodilation. So that means that the blood vessels in our periphery are going to expand, which brings more blood to the surface, so that way we can cool down. It also allows more blood flow to the skin, and therefore the sweat glands become activated. So sweat glands start to secrete their product, which also helps us to perspire and have evaporative cooling. The respiratory centers are also stimulated, and the depth of our respiration will increase. Now contrast this with when we want to conserve heat when we're cold. So our sympathetic vasomotor center is going to decrease the blood flow to the dermis, which is the exact opposite. So it's going to constrict all those blood vessels, so that way we shunt blood primarily to keep our organs warm. And that's all. And then also we're going to reduce heat loss by radiation, convection, and conduction. So the blood that returns from the limbs is going to be shunted to the insulated deep veins of our body, and that way the heat is conducted from the warm blood in our arteries out to the cooler blood that's out in our veins. So we have what's called a countercurrent exchange. So this traps the heat that we have, the little heat that we have, close to our body core and reduces losses, which is important because if your hands are cold, it really doesn't matter. Your hands can go on and survive. But if your brain gets cold, if your lungs get cold, if your heart gets cold, we have problems. So we have to keep all of our internal organs, specifically our vital organs, warm. So this process keeps our core body at a normal temperature so we can have normal thermic reactions. The main purpose of the heat gain center in the brain is to prevent hypothermia, which is just below normal body temperature. And so let's discuss some of these mechanisms for promoting heat gain. So when the temperature drops, the preoptic area of our hypothalamus is going to recognize this. And as a result, two things are going to happen. We'll have our heat loss center inhibited and our heat gain centers activated to make sure that we are able to warm up. One of the ways in which we generate heat is by shivering thermogenesis. And this is just shivering, like exactly what you think it is. So what happens when we shiver is we're increasing muscle tone throughout our body, and that increases the consumption of skeletal muscle energy and produces heat. So we involve both agonist and antagonist muscles in shivering, and the degree of the stimulation is going to vary depending upon the demand. But if your heat gain center is extremely active, you will begin to shiver. And that shivering is going to increase the rate of heat generation up to 400%. So it's actually a highly effective mechanism to generate warmth, even though you wouldn't think that shivering really does anything. Because if you're like me, anytime you've ever shivered, you probably thought it was just purposeless and didn't do anything to warm you up. But it actually, in fact, does considerably raise your body temperature. Maybe not enough to have you feel comfortable, but enough to help keep your internal organs at a euthermic temperature. In addition to shivering to create heat, we also have non-shivering techniques. So we call these non-shivering thermogenesis. And what it is involving the release of hormones throughout your body that are going to increase the metabolic activity of all of your tissues. And keep in mind, the byproduct of metabolism is heat. So the heat gain center in our hypothalamus is going to stimulate the adrenal medulla. And the adrenal medulla is going to cause the release of epinephrine, which then immediately makes you feel extremely alert, for one, and then also increases the rates of glycogenolysis and metabolism. In children, if they have low body temperatures, that can stimulate also the release of other hormones, for example, TRH and therefore TSH, which is thyroid stimulating hormone. So the results will be that you have a release of thyroxine, which will then increase the rate of catabolism, but it does take days to weeks for this to work. Thermoregulatory responses vary both among and within individuals, and this can be due to a number of things. The first is acclimatization. So this just means that if you're in a very specific environment, over a period of time, you will have physiologic adjustments to allow yourself to accommodate that environment. The other thing is variation in body size. So surface to volume ratios will decrease as size increases. So what that really means practically is that smaller individuals are going to be able to lose heat much more easily than large individuals do. We discussed brown fat previously in our tissue lecture, but just to discuss it again, brown fat is thermogenic tissue, and it's not the same as our typical white fat that we have in our abdomen. Brown fat is typically found in infants and also in hibernating mammals. This tissue is really important because it is thermogenic, meaning it creates heat. So it is rich in heme, cytochromes, and also mitochondria. And that mitochondria, of course, is very important because as the powerhouse of the cell, mitochondria is responsible for creating ATP. So having cells that have a lot of mitochondria mean they can generate a lot of energy. So fatty acid catabolism is going to release heat, and that radiates into the surrounding tissues and into the blood. So these temperature regulating mechanisms aren't really fully functional in infants, and they can't shiver. So this is an important reason why we have brown fat infants, because they do lose heat quickly because they're so small, and they really need to be able to hold on to their heat. Otherwise, their organs, of course, would fail. So in adults, you may find some of this, but not a lot, around the neck and the upper chest and shoulders. Thermoregulatory responses in adults vary considerably, and I've already discussed some of these. So they vary according to body mass, so larger individuals lose heat more slowly than smaller individuals. And tissue distribution, so more adipose tissue acts as an insulating effect. And people have different settings of their hypothalamic thermostats. So if you've ever gone shopping for a backpacking or outdoor sleeping bag, you'll find that they're rated for different temperatures. And they're also rated for men and for women because typically, this is just a finding that people have noticed over time, men seem to sleep colder than women. So women want to be warmer when we sleep, which I think is kind of interesting. Anyway, individuals with more subcutaneous fat are going to shiver less than thinner people, which is an interesting side effect. And body temperature is going to vary daily, so it has these daily oscillations up and down. The ovulatory cycle in women is going to cause temperature fluctuations considerably, especially with a spike right around the time of ovulation. Fever is defined as a body temperature that is greater than 37.2 degrees Celsius or 99 degrees Fahrenheit. Now, I know you're probably familiar with using Fahrenheit and the imperial system in general. For example, I'm Canadian and I still use the imperial system to say I weigh X number of pounds, not X number of kilograms. But nonetheless, when you are charting and working in the hospital, you will be using the metric system. So get used to using that now. So 37.2 degrees Celsius is considered effectively a fever. So, but for a lot of reasons, this isn't always a disease process or considered pathological. So in young children, they can get fevers that come and go. It can result from just exercising in warm weather. So if they get just a little bit warm, they can cool down again, sometimes quite quickly. Fever can also result from heat exhaustion or heat stroke, congestive heart failure, conditions that impair your sweat gland activity, and also circulating pyrogens. Figure 25-14 in your text shows you how vascular adaptations for heat loss and conservation work. So in A, over to the left, you'll see that the more peripheral blood vessels are going to be vasodilated in order to allow more blood to come to the surface for radiative cooling and also to stimulate our sweat glands to allow for perspiration and therefore evaporative cooling. In the cold environment, what we have is instead, those peripheral vessels will instead vasoconstrict, so that way there is less blood that's getting exposed out to the surface to be cooled. So we keep the warm blood towards the core of our body, both in the core of our arms and back to the core where our organs are. And then in image C, you can see what a countercurrent heat exchange looks like as the heat radiates from the warm arterial blood into the cooler venous blood flowing in the opposite direction.